All right, welcome to this lecture. We are still looking at pharmacodynamics. And as you remember, last time we ended at words like potency, things like efficacy. That's where we, that's where we ended. And we want to continue or proceed from there. And maybe just to remind us a little bit, we say that um, potency is the amount of drug that is basically needed and elicits a response or a maximum response, while uh, um, efficacy is basically the response that a drug is supposed to make. And with efficacy, there is binding, as we said earlier on, but at the same time, it makes an effect. So with efficacy, there is binding, which is affinity, and then we also have an effect. And so we also differentiated between affinity and potency, rather affinity and efficacy, where we say that with affinity is basically the ability of the drug to bind. But if it goes ahead to create an effect, then we refer to that as efficacy. Right. So we want to continue from where we ended. We had mentioned that in that diagram, as you see, we are seeing that morphine has a higher potency than pyridine. However, they are both efficacious. On the other hand, if you compare aspirin and codeine, we are seeing that codeine has a higher, it is more potent, and also it is more efficacious compared to aspirin. And that is actually why aspirin is being referred to as a mild analgesic in this diagram, while codeine is being referred to as a moderate analgesic. All right. So in that slide, we are comparing relative potencies. You can see hydromophone. You're seeing morphine, you're seeing codeine and aspirin. Yeah. So we are seeing hydromophone. We are seeing morphine. We are seeing codeine as well as aspirin. Aspirin. And if you look inquisitively at the diagram, it is basically telling us that basically hydromophone is more potent compared to morphine. But if you can see on the diagram, they basically are similar indications. On the other hand, if we compare aspirin and codeine, again, we can see that codeine is more potent and more efficacious compared to aspirin. And so that is basically a chart that is depicting the relative potency of these drugs. All right. So let's talk about what we call agonists and antagonists. Yeah. So an agonist, as we said earlier on, it has both affinity and intrinsic activity, or both efficacy and affinity. So and you remember what we said: if a drug has both, then it basically binds and will therefore also have an effect on that drug. Yeah. So we are saying that an antagonist, on the other hand, has only affinity. In other words, it only binds, but it does not cause an effect. In other words, it basically just blocks the receptors, but it does not have any effect whatsoever. So it it basically blocks the receptors. And we also have what we call partial agonists. This is they basically have affinity and have less intrinsic activity because they are partial agonists. Yeah? And then so with partial agonists, if we have a competitive antagonist, it basically can overcome a partial agonist. And if you draw, if you can see that graph there. If you draw a log of concentration of a drug versus the percentage, yeah, or the maximum effect, yeah. If you draw something like that, if you have a chart, if you have something a chart like that, so if we are to consider a drug or an enzyme, yeah, if it's given orally, it basically stuff like that, and then it basically goes like that, yeah. And so if we give an agonist X, yeah, if we give an agonist X, it basically means that the drug, you know, will be shifted to the we call this the right, yeah. So it is shifted to the right, as you can see in that diagram. Yeah. So if we give if we have our drug X here, if we have our drug X, and then we give an agonist, and rather an antagonist Y. Yeah. Maybe you should know that, or maybe the first thing that you should know is that an antagonist basically blocks the activity of an agonist. Right? So imagine this was X was our initial agonist that was given, yeah, and for it to create a maximum response. This is you know, this is the concentration of the drug that is given, yeah, for it to create a maximum response here. Right? But if we give an antagonist, and in that case a competitive antagonist, right? So we see that the reaction, this is basically a reaction. For us to see a maximum response, of this drug here, of this drug, you see that you need to increase the dosage, kind of like increases a little bit. The dosage increases a little bit, as you can see from this diagram, yeah, according to maximum response rate. This was the original dose, yeah, maybe D1, and then this becomes D2. Yeah, the dose that you need to give for a drug, for the initial drug or for the agonist, to basically bring about a maximum response. Yeah? So with successive giving of an antagonist, basically the graph or the reaction is given to the right, as you can see in that diagram. Yeah? So we have an agonist plus raising a competitive antagonist, Y. It basically drives the equation, or it drives the reaction on the other side. What does it mean? It basically means that for us to achieve a maximum response using our drug that we were giving initially, we need to increase the dose. That is basically what it means, because it is basically competitive. Yeah? It is basically competitive, and of course that comes as a result of just imagine, maybe if we can draw a diagram. Yeah, we have assuming this is our receptor, and this is where the drug has to attach. This is our agonist. That is our agonist, and this is our drug D. This is our drug D, And then we also have an antagonist, a competitive antagonist, Y. Yeah, we also have a competitive antagonist, Y. So it basically means that if we increase this drug E, it will basically overcome this, because they're kind of like in a competition. Yeah, they are competing. Yeah? So for us to achieve maximum response, we really need to increase the amount of this drug of ours, so as to achieve a maximum response with the competitive antagonist. Yeah. So basically that is what is referred to as an agonist and an antagonist. All right, so agonists, these are basically drugs that interact with and activate receptors. Yeah, they possess, as we said, they possess both affinity and efficacy. They are basically of two types. We have what we call a full agonist. Yeah, so these basically produce a maximum. Yeah, they produce a maximum response. Yeah, like for example, they give us a maximum response. And then partial agonists basically they don't give us a full response. So this can be partial, while if this is response, if this is a response, this can be partial, this can be partial, this can be a full agonist. So it is all about response, or what we refer to as efficacy. This is what differentiates these two drugs as either full agonist or partial agonist. All right. So continuing from there, as we can see, as earlier on seen, a full agonist, if this is response and this is the dose, for us to get a maximum response, rather for us to, if we get a maximum response, a high response, then we refer to that as a full agonist, compared to what we refer to as a partial agonist. Now, on the other hand, an antagonist, these basically interact with the receptor and therefore bind. So for that matter, or for that case, they have what we refer to as affinity. However, they do not have efficacy. They do not create an effect. Their role is just to bind. Their role is just to bind. Assuming this is our receptor, maybe that is where the drug has to bind. So if it is an antagonist, for it just binds in the space there. It does not allow any other molecule or any other drug to bind there. Yeah. So it basically just binds and it seals off the space. It seals off the space. And therefore, other agonists or other drugs are not able to come and bind to the receptor. Yeah. They're not able to come and bind to the receptor. And so, for that matter, or for that case, we say that an antagonist has affinity, but not efficacy. Yeah, and of course, there are two types of antagonists. We have what we call competitive and non-competitive. Yeah, we have competitive and non-competitive antagonists. Yeah? So what is a competitive antagonist? So a competitive, this one basically, just like the word competition, it competes with the agonist for the receptor. Yeah. So if this is the receptor, if this is our receptor, and this is the place where either the agonist or antagonist should bind. What actually happens is that if we have if we have our drug here, D, and we have our antagonist here, maybe A, yeah? So both of them, both this and that, they compete for this site. They compete for this site, and therefore are referred to as, competitive antagonism. Yeah, competitive antagonism. And what does that mean? It basically means that since they're in competition, they're increasing, whichever it can basically surpass or displace the other. Yeah, increasing whichever of these it helps to displace the other, and it basically shifts the reaction to it shifts the reaction to the right, yeah? And therefore it reduces the affinity of when you increase the antagonist, the affinity of the receptor for the drug basically reduces. Yeah, because you have a lot of the antagonist in place. So that is what we refer to as a competitive antagonist. On the other hand, a non-competitive, this one stays bound. Yeah, it stays bound to the receptor, and when it binds to the receptor, again, I'll give you this as an example. Yeah, I'll give you this as an example. Assuming this is our receptor, R, yeah, and then this is our drug. This is our normal drug. This is our normal drug D. And so we can have our antagonist. Our antagonist can even assume any shape because it is basically non-competitive. So it can assume any shape. It can be probably like that. Yeah. And so this antagonist, which is non-competitive, can bind. Yeah. It does not necessarily need to bind in this place here. It can even bind here. Yeah. It can bind somewhere there. And once it binds there, then this receptor loses interest for the drug. Yeah, it loses interest. In other words, its affinity decreases for the drug. Yeah. So this competitive, non-competitive antagonist can bind anywhere, and once it binds, it basically blocks, or it blocks it actually does not block, but it reduces the affinity of this receptor to that drug. Yeah? And for that matter, it basically binds it binds anywhere on the receptor. Yeah? And so it binds irreversibly. So once it binds here, it is irreversible. So it basically means no matter, even if you increase the drug of choice, even if you increase the concentration of the drug of choice, it does not really matter because it is binding irreversibly. And then, of course, it produces a shift, yeah, of our curve as we saw. And most of the curve for competitive antagonists and whatever look almost the same, but as more receptors are bound, essentially destroyed, the agonist becomes incapable of eliciting what we call a maximum response. This is what I was saying about that. As we have more of these binds, then we shall no longer have this drug eliciting a maximum response here because it is being non-competitively antagonized by this, by this drug or by this antagonist. All right. So that is basically what we heard as far as agonists and antagonists are concerned. Yeah. Now the other issue that you need to know is the term That word is very important. The term desensitization. The term desensitization. So that word is actually very important. And what does it mean? That you know, once you have a lot of agonists, once you have a lot of drugs going to that receptor, yeah, very soon, the receptors lose what we call interest in the drug, yeah, in the drug, and as a result, we refer to this as desensitization, yeah. And so for you to basically achieve a maximum response, sometimes you need to either increase the drug or or you will not achieve a maximum response. And what actually happens is that uh, basically, because of desensitization, two things can happen. One is that the receptors where the drugs are supposed to bind basically decrease in number. Yeah, so you have a decrease in number, might be a cause of decrease in number of receptors, or it can be as a result of a decrease in what we call signal transduction. Yeah? We are going to look at that, signal transduction. So it can be as a result of either this or that, and all that produces desensitization. And by desensitization, we mean that you will not achieve the response that you actually are interested in. Yeah? The drug will not achieve the response that is due because of this desensitization. Yeah? So in other words, when we ever give a drug, a time comes when like example, when a drug is when someone is so much maybe on antihypertensives for years and years, the time comes when the receptors decrease. Yeah? When the receptors for that that particular drug decrease, or signal transduction decreases, and as a result, the patient will not get an effect that is basically intended. But we also have antagonists usually tend to upregulate receptors. In other words, once we give an antagonist, usually we have more manufacture of receptors. Remember, we said receptors are proteins. Yeah, receptors are basically proteins, so they can be manufactured anytime, or they can be destroyed anytime. So antagonists basically tend to bring about upward regulation of receptors, while agonists basically try to bring about what we refer to as desensitization, or what we call a decrease in number of receptors, or a decrease in signal transduction. Okay, basically these are curves that are showing us those response, what we refer to as those response curves. But maybe before we go, we come back to that. Let's first we need to know what we mean by effective dose and lethal dose. Now, for any drug to be given, yeah, for any drug to be put on market per se, then lab studies are done on individuals, and also on on probably rats, and so that we determine what is the effective, yeah, effective. There's what we call lethal, and then toxic doses. Yeah, so these are doses. So once you see ED50, TD50, LD50, that basically means that the effective dose, 50% of the population, lethal dose, you know. So what do these figures mean? Effective dose of what we call ED50 is basic. As I said, or as I mentioned earlier on, when a drug is put on market, for a drug to be put on market, it basically calls that you know, they know how much should be given, yeah, for this drug to actually work. Like, for example, if this drug is going to be maybe if it is intended to maybe if it is an antibiotic, you need to know how much of this antibiotic should be given to have, so as to have you know, an effective, for it to be effective, in order to create a maximal response. Yeah, and so that is where we have these things like lethal dose and toxic dose and 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 what the toxic dose, lethal dose, and effective dose. Yeah. So effective dose is basically is the dose at which 50% of the population of sample manifests a given effect. Yeah, and usually it is shown in terms of the curves, as the curves that you have seen. Yeah, those curves there. Yeah? But we are saying that effective dose is that dose that you usually give in 50% of the population, and then it works. It works. Yeah, it basically works. It takes away maybe it takes away the problem that you have. If it is a pain, you know, if it is maybe an infection, you know, so how much you should give in 50% of the population. That is what we refer to as effective dose. And then toxic dose 50, this one is the dose at which 50% of the population manifests what we call the toxic effects of that drug. Yeah, that is what we call the toxic dose. And then the lethal, basically 50% of the patients being killed. Yeah, being killed or the subject. Usually we don't refer to patients per se, but because sometimes it's not tested on human beings. Yeah, it might be tested on animals, rats, you know. Yeah, but lethal dose is the killing. Yeah, so that dose at which 50% of the subjects basically die at that particular dose. Now, what is the importance of these doses? The importance of this dose is because they are important to determine what we call the therapeutic index or the therapeutic dose. Yeah? And of course, the therapeutic index, as you can see in that diagram, is basically given by effective, rather, it is given by toxic dose or lethal dose divided by toxic dose 50 or lethal dose at 50 divided by effective dose at 50. That gives us our therapeutic index. And if you still remember very well, we said the therapeutic index is the range with which you can, you know, give a medication, and this is basically how it is determined. Yeah? Now, compare, if you're to compare a drug A, as you can see there, 50% of the drug being effective, of bringing bringing about its effects, and then the effective dose is basically that, as you can see, and then the lethal dose is at 50 is that which is given, rather which causes patients or subjects. And if you can see with that one, you still have the same diagram, but this one, as you can see, with this drug A, with that drug A, you can see the therapeutic index is small compared to. In other words, the difference between the effective dose and the lethal dose is small. Yeah, if you observe if you observe them, compared to this one, where we have the difference or the space between the effective dose and the lethal dose is quite big. So I would pose a question: drug A and drug B, if both of them were intended probably to bring about the same effect, like maybe if both of them were painkillers, which of those two would you prefer using? And of course, the answer is yes. The answer is B. Yeah, the answer is B because it has a larger, therapeutic index. As we talked about this, so we are saying the higher the therapeutic index, the better the drug. Yeah, and basically they vary from one, like for example, for some kinds of drugs, and then to greater than 1,000. Yeah? So drugs acting on the same receptor and enzyme system often have the same therapeutic, index. Yeah? Basically. Now, it is also very important to consider what we call what do you consider before you give a particular medication. Yeah? All this is still covered under pharmacodynamics. Yeah. Now, dosing consideration. Dosing consideration. Now, what do we mean by dosing consideration? We basically mean those things that should be at the back of your mind. Those things that should be at the back of your mind as you give a what? As you give a drug. Yeah, as you give a drug. What are those things that should be at the back of your mind as you give a drug? Now, one of them and very most important is age. We talked about this earlier when we were talking about issues that have to do with the elderly and the neonates, because livers and kidneys are still growing for the neonates, while for the elderly these things are aged. Yeah, so basically you need to consider that in consideration. Gender, pre-existing pathological condition. Like, for example, there are some drugs that are contraindicated in people who have renal condition. Yeah? Or there are some drugs that are contraindicated in people who have liver problems. Yeah, so the pre-existing pathological condition of the patient is very important. Psychological factors, placebo effect. By this we mean that sometimes a patient might not necessarily require a particular drug. Yeah, but once you give them what we refer to as a placebo, then they are assumed to have their pain gone. Yeah? So psychological factors are also very important. Stress and emotional state. Drug tolerance is basically the ability of someone to withstand or withhold a drug. Yeah? And then what we refer to as drug-to-drug interactions. There are some drugs that are not given in combination or given carefully or put in some considerations before you give the other drug. Like, for example, if you're to give maybe iron with some other drug, it is basically recommended that you basically give the drug first, maybe, and wait for about 30 minutes before you give iron. So and all this is intended to avoid or prevent drug interaction. Yeah? So basically, that is very, very important. Now, let's talk about in this last presentation we are going to talk about what we call receptors. Yeah, receptors, and for that matter, what we refer to as signal transduction. Signal transduction. Now, when a drug binds to a receptor, yeah, that is basically not it. Yeah, it's not just that only. Yeah, what actually happens is that once a drug binds to a receptor, several signals, yeah, move along the receptors and for that matter, cause or bring about an effect. Yeah. So there are types of receptors that we are going to talk about, and as you can see, we have what we call enzyme-linked receptors. We have what we call ion channel-linked receptors. We have G protein-linked receptors. We have nuclear, linked receptors. Yeah, and so we are going to talk about those now. Which basically this shows us, or this tells us that drugs have different receptors. All drugs do not basically, or do not necessarily have the same receptors. Yeah, but drugs have different receptors, and so there are those that are enzyme linked. There are those that are ion channel linked. There are those that are G protein coupled. Yeah, or G protein linked receptors, and there are those that are nuclear. So in other words, if it is nuclear, it basically means that the drug has to traverse the plasma membrane, and then it goes into the cytoplasm up where the nucleus is. Yeah, so that basically means that that is where the receptors of that drug are. So in other words, while it is traversing the cell membrane, no effect until it reaches the nucleus, until it reaches the nucleus. So these are the ones that we are going to be talking about. And as you can see in the diagram, with the enzyme linked, it basically has an enzyme on top, which basically causes modifications once the drug binds to the receptor. We have ion linked. You are very well conversant. You've heard about these ion channels, ion gated channels. Yeah. So you're very, very well conversant with that. And so if you can see, or if you look on the diagram, it is very clear that you have ion channels through which, of course, if a drug comes and binds to the receptor on an ion channel, then it causes several series of reactions, as we are going to see. And then we have G protein coupled, which basically ends up producing what we call cyclic AMP. Yeah, and then we also have what we call the nuclear, or what we call the gene linked receptors. All right. So starting with what we call the G protein receptors, as you can see in the diagram. So what is the structure? It is basically like it is a polypeptide chain, yeah, that is basically made up of seven helices, as you can see in that diagram. Polypeptide chain, it's a chain of polypeptides, and they're basically 7 in number. And as you can see on that diagram there, there is a place where the drug is supposed to bind, as you can see. And then there is a G protein attached to the cytoplasm side, to the cytoplasm side of the membrane, and then of course it functions as a switch. Yeah. So when the drug binds to the membrane, to the upper part, to the receptor, then the G protein basically acts like a switch to cause other changes that occur within the cell to bring about several other changes. All right, so this is basically what happens, as you can see. We have a chemical, when a chemical signal binds, yeah, as a ligand to a G protein linked receptor, what actually happens is that the G protein linked receptor changes shape, yeah, and interacts with the G protein, as you can see in that diagram there. Yeah. So we are saying that once the drug binds here, initially the diagram is like this, as you can see. But once the drug comes and binds, so you have the G protein linked receptor changing in shape, yeah, and then interacts with the G protein. So you can see initially they were separate, but then when this receptor binds, then the G protein basically interacts with the receptor to cause a series of reactions. So from there, so interaction basically causes GDP, yeah, to be displaced, and GTP to be bound to the G protein. In other words, you have that interaction and that reaction basically creates energy, or it brings up creates energy of an extra phosphate, which links to the GDP to basically produce to bring about what we call the GTP. So the active G protein then binds together, binds to another protein, usually an enzyme. As you can see here in this, so we have the extra phosphate. Because of the reaction, we have an extra phosphate bringing or giving us what we call GTP, or what we call guanosine triphosphate, which binds to the enzyme, yeah, which binds to the enzyme, yeah, as you can see here. And then once it binds to the enzyme, then we have a series of other reactions which basically lead about or bring about, cellular response. And so cellular response can be in different forms. It might be maybe pain relief. It might be that these cells need to manufacture or make more proteins, yeah, protein synthesis, and for that matter, yeah. So different cellular responses happen, and as a result, the drug basically produces its effect. So the enzyme, once the enzyme is activated, then the G protein hydrolyzes GTP back to GTP, and then basically it returns back. The G protein releases from the enzyme, and the reaction stops. So it returns back to normal. It returns back to normal as it was. So the point to take home here, ladies and gentlemen, is that we have different drugs have different receptors, and we say iron-gated, G protein, nuclear, and among others. So basically four. Now we started with what we call the G protein linked receptors. Yeah. Now the G protein linked receptors, where most drugs fall, yeah, they basically have it's a helix of polypeptide chain, 7 in number. Yeah. So as you can see, it traverses. It has a part within the membrane, and a part, inside the membrane. Yeah. So the part outside the membrane is where we have our receptor. Is where we have the receptor. In other words, is where we have a point where the ligand, or where we have the drug coming to bind, as you can see in that diagram. Yeah. So, and once it binds, basically it changes the structure of that G protein. Yeah, it changes the structure of that G protein, and bringing about extra energy, which causes transformation of the GDP to GTP. In other words, we have an extra phosphate that gets bound to the GDP to create or bring about GTP. And as a result, because of that energy, it forces the GTP to even go and bind to the enzyme, yeah, to the enzyme, or what we call the intramembrane enzyme, usually protein. And once it binds to that one, then we have a series of cellular responses, or a series of reactions that might be required for us to get a response. However, it does not continue forever. No, it does not continue forever. There comes a time where things come back to normal, or where maybe the drug is no longer, its concentration is no longer effective. What actually happens is that the drug will now displace off the receptor, and things will turn back to normal, as you can see in the diagram above. Yeah, so that is basically the G protein receptor. Yeah, the G protein linked receptor for that matter. And then we have another one which we refer to as the tyrosine kinase receptors. Yeah, tyrosine kinase receptors. These are basically receptors that exist as individual polypeptides, and each has what we call an extracellular signal binding site. Yeah, so it has an extracellular outside the cell, extracellular signal, and it also has an intracellular tail. In other words, there is a communication from the outside to the inside. There is a communication. So an intracellular tail with a number of tyrosines and a single helix spanning the membrane, as you can see here. So that is basically the receptor, tyrosine kinase receptors. And so what actually happens, yeah, is that when the ligand binds to, the ligand should bind to both receptors, yeah? And then what actually happens once the ligand binds, so once there is binding, there is some form of energy that is created that basically causes or brings about transformational changes, yeah? So once we have binding of the ligands to these two positions or to receptors, then the receptor polypeptides, you know, aggregate and therefore form what we call a dimer. In other words, initially they are separate, but once we have binding of the ligand, then they bind or then they combine together to form what we call a dimer. And all that happens because of the extra energy that is basically in use or that is basically produced. And that is what we refer to as signal transduction. Yeah. So now, this basically activates the tyrosine kinase parts of the D-dimer, and each phosphorylates using ATP the tyrosines on the toe, or rather on the tail of the other polypeptide. Yeah, as you can see here, we are seeing that initially we had the tyrosine. Yeah, so once we have binding on both, they bind, and then they basically produce a dimer, and as a result of binding, there is phosphorylation of each tyrosine kinase, yeah, of each tyrosine. There's phosphorylation of each tyrosine, producing what we call tyrosine phosphate, as you can see in this. So the receptor proteins are now recognized by the relay proteins inside the cell. And as a result, relay proteins bind to the phosphorylated tyrosines and therefore produces different cellular responses. Yeah? So in other words here, just like we had G protein coupled, this time we have tyrosine kinase receptors. Yeah? And of course, the receptor should bind on both. The ligand should bind on both, and once it binds on both, because of the energy that is created, then they bind together, and as a result, when they bind together to form a D-dimer, each phosphate using ATP basically phosphorylates you know, the tail of the tyrosine, yeah, forming a tyrosine phosphate, which tyrosine phosphate is now active to bind to the enzymes, as you can see the enzymes here, and then basically produce or bring about cellular responses. So basically that is the tyrosine kinase receptors. Now we also have what we call the ion channel receptors, or what we refer to as the ion-gated receptors. As you saw earlier, it basically looks like that in the diagram. And so there is a point, or there is a place where the drug is supposed to bind. Yeah. So if you see binding here, once it binds, then it opens. Yeah. It opens, and then the signals are able to go through. Yeah? So what actually happens, then the signal molecule binds as a ligand at the specific site, and what actually happens is the transformational changes open the channel, allowing the flow of ions into the cell, and then the change in the concentration within the cellular cell basically triggers or brings about cellular responses. Yeah. So those are what we call the ion-gated or the ion channel receptors. Now, I'm going to give you an assignment. tell me which drugs, give me examples of which drugs use which particular receptors. With examples, tell me which kinds of drugs, what types of drugs use what kinds of receptors. Yeah, that is the question that I'm giving you. Then we also have what we call intracellular receptors. So these ones, as the word states, intracellular, the receptor is on the nucleus. The receptor is on the nucleus. So the drug will not produce any changes outside unless it reaches that, unless if it goes to the nucleus. And therefore, there should be means or ways of how the drug should get inside the cell. Yeah. So not all signal receptors are located on the plasma membrane. Yes, some are proteins located in the cytoplasm or nucleus of the target cells, and then the signal molecule must therefore be able to pass through the cell membrane. Yeah, and then once it passes through the cell membrane and then it meets the receptor on the nucleus, then it causes or brings about several changes. Like, for example, nitric oxide, which is known for vasodilatation, it has its receptors on the nucleus. And then other examples, most steroids, yeah, most steroids also have their receptors on the nucleus. By the way, that is why their effects are quite long. Yeah, that is why their effects are quite long. Yeah, because they have their receptors within the nucleus. All right, so let's talk about what we refer to as second messengers. Second messengers, these are basically maybe I should tell you that once a drug binds to a receptor, yeah, like for example, let's take an example of the G protein link. Once that drug binds to the receptor, yeah, it is not the one that basically will produce or will cause all the changes. Yeah, but once it binds, energy is created, and at the same time, what basically causes or what will basically bring about the different cellular changes that take place is what we call the second messengers. Yeah, the second messenger. And so what are second messengers? These are basically small non-protein water-soluble molecules or ions. Yeah, and then they rapidly spread throughout the cell by diffusion. Yeah, and the two most common second messengers are what we call cyclic adenosine monophosphate, or what we call cyclic AMP, and then calcium ions. Yeah, so especially, like for example, with G protein, yeah, the second messenger is cyclic AMP. It is the one that basically the one that is produced, and once it is produced, then it disperses throughout the cell to basically bring about changes. So calcium ions and the inositol phosphates, calcium is more widely used than cyclic AMP, and it is used in neurotransmitters, growth factors, and in some hormones. So those drugs that are hormones, those drugs that are growth factors. So for them, they use calcium ions as their second messengers. Yeah. So what actually happens is that it basically increases. It causes an increase in calcium ions within the cell. Yeah. So that drug brings about an increase in calcium ions within the cell, bringing about several effects, like maybe muscle contraction. You have done this in physiology, how a muscle contracts. Yeah, the sarcoplasmic reticulum releases too much calcium. Yeah, so so once this calcium is released, then you have muscular contraction, among other secretion of certain substances, and then cell division. Yeah, all this is as a result of calcium that is being released as a second messenger. Yes, into the cell. Yeah, and basically, what are the benefits of this signal transduction pathway? is that one, they cause signal amplification, and two, they bring about signal specificity. Now, what is signal amplification? Is that they cause things to be amplified, yeah, to be amplified, yeah. And so proteins persist in active form long enough to process numerous molecules of substrate. So each catalytic step activates more products than the one preceding it. And so we have amplification of a process of whatever process that we are intended or we are interested in. And so that is basically signal amplification. Okay? So this diagram basically gives us it gives us a summary of how cyclic AMP, or of how a second messenger cyclic AMP is created, and how it basically brings about amplification. Yeah. So they give an example, like for example, epinephrine. Yeah, you know epinephrine. Epinephrine basically binds to a G protein linked receptor. It basically causes activation of the G protein, as we saw. It produces active adenyl cyclase, which active adenyl cyclase basically produces cyclic adenosine monophosphate, and then the reactions basically continue. But in this chart, what you should know is, in this particular context, the second messenger, or what is actually creating or bringing about different or several effects, is cyclic AMP, which is created as our second messenger. Yeah. So, in summary, most drugs act through receptors. Of course, not all drugs will act through receptors, but most of them. So, I want to give you another assignment. Give me examples of drugs that do not use receptors. Yeah. And there are four common signal transaction methods, or four common receptors, as we saw. And then the interaction between a drug and receptor can be described mathematically and graphically. We saw affinity, we saw antagonism, and we basically talked about desensitization and sensitization of receptors. So that basically marks the end of our pharmacodynamics. Yeah. If you have any questions, please do not hesitate to ask. I'm also going to avail you with recordings with lectures of the sympathetic and the parasympathetic nervous system. And when we meet next time, we shall be talking about the distribution of these in the body. Yeah. So basically, this is the recording that marks the end of our pharmacodynamics. Please listen to it over and over again, as so as to understand. But also, in case you have any more questions, then feel free to ask when we have our live sessions on Zoom. Otherwise, thank you so much. Stay home and stay safe.